Thanks very much, Carol, for that introduction. And I'd like to thank everybody for coming here and allowing us to share with you some of the exciting work that's been happening in this space. So, you know, I think we've heard a lot about cancer immunotherapy in the last few years, and there's been a lot of excitement, and I think that excitement is quite rightly justified. And the developments are happening so fast that it's very hard to keep up with them, and it's certainly a real challenge for us in the lab as well as in the clinic. So what I want to do today is actually just give you a brief history of cancer immunotherapy, and just to place it in context, and just to get everyone to appreciate the pace at which all this is happening. So as Carol mentioned, just got to work out how this works. Yeah. So my name is Tri Phan. So I run the intravital microscopy lab, and one of the things our lab's really interested in is trying to understand how immune cells interact with cancer cells, and we do that through a process using a very expensive piece of equipment called a two-photon microscope. And really, I wouldn't be here to talk to you about today without the help of all the supporters, because that microscope was funded by the Garvan Research Foundation. So cancer was, the term was actually originally coined by Hippocrates, who called it karkinos. And one of the reasons why he called it that was it had, so karkinos is the Greek word for a crab. And so the cancer, it was locally invasive, and it would spread out and would stretch its claws, so to speak, into the surrounding tissue like a crab. And cancer tissue is also very hard, like the shell of a crab. And in fact, cancer is such a devastating disease that it led this oncologist, who wrote a best-selling book, to call it the emperor of all maladies. And it's a devastating disease that, despite all our medical advances, still afflicts a large number of people and causes incredible mortality and morbidity. So, in fact, although the word cancer was only described a couple thousand years ago, it turns out that actually cancer's afflicted the human race for an incredibly long period of time, and even afflicted our early ancestors. So if you go back to this fossil, which is dated to 1.7 million years old, this is a bone from the foot, and there's this cancer growing out of the bone, and this is what we would now call an osteosarcoma. So in this micro CT scan sectioning on the side, you can see the typical cauliflower outgrowth of this cancer. So this is something that we as a species has been dealing with for a long time. But the story over a long period of time has not been a happy one. So in fact, if we go back and look, so here is a mummy that was in a museum in Lisbon in Portugal. And when they did a scan of the mummy, what you can see, and so here are the vertebrae in the spinal cord, all these spots, which are the cancer from the prostate of this mummy that spread to the bone. So cancer is locally invasive, and it's spread to the bone, and this is something that we've dealt with for since Egyptian times. So this is the first, I guess one of the first medical textbooks that was attributed to Imhotep, and in it contains descriptions of surgical procedures, mostly orthopedic, but amongst the things it described was a breast cancer. And there's a line that struck and resonated through the centuries, and that is that for cancer there is no treatment. So we come from a very nihilistic period of time in which cancer was something that was regarded as a death sentence. This idea persisted through Roman times. So this is a very famous Roman physician who wrote one of the first, in fact, one of the first encyclopedias. And in this text de Messina, he described that even after excision, when a scar has formed, nonetheless the disease has returned. And this describes this other problem that we have with cancer, and that is that even though we can resect the primary tumour, we can make the cancer go away, after periods of time, the cancer comes back and the disease relapses. And in fact, relapses is a major cause of mortality and morbidity even to this day. So is there hope? Well, I guess it's fitting that at the Garvan Institute, with its religious connection, one of the first signs that there may be a way back is this gentleman here. This is Peregrine of Laziosi. So he was a very devout man, and he would kneel down and pray every day, and as a consequence of that, he developed what probably were varicose ulcers on his legs. And so these ulcers are, over time, because of the constant inflammation, will give rise to a cancer, what's called a Magellan's ulcer. And so he developed a cancer in his leg, so that's shown here. And it actually became so bad that he thought he was going to die, and that the only option left, which was the treatment at the time, was the local barber or surgeon had planned to actually amputate his leg. So the story goes that he actually, despite all the pain and suffering that he'd gone and had accrued to him from kneeling, he knelt down for one last time to pray to God for a miracle. And as it turned out, what had happened was that because the ulcer had become infected, it actually became so foul-smelling that the surgeon actually couldn't come very near it. But overnight, the ulcer healed and he was cured. And that was proclaimed as a miracle, and he was made a saint. And he's actually now the patron saint of cancer. But this story actually illustrates a really important point, and that is the immune system can be actually a double-edged sword. We know that under some circumstances, because of chronic inflammation, the immune system can actually give rise to cancers. We know that, for example, in some of the colon cancers, and we know that in the case with St. Peregrine, with these varicose ulcers in the leg. But at the same time, even though it can give rise to the cancer, it can actually kill the cancer. And what we think has happened here is that because he developed an infection in the wound, the immune system became activated, and in the process of getting rid of the infection, it also turned its attention to something that it overlooked and realized that the cancer was something that needed to be dealt with, and it eradicated it. So this actually was the first recorded case of spontaneous regression of cancer, and this, to us, is really the shining light that tells us where the future might go. Over time, it took a long time for people to recognize the importance of that description of the spontaneous regression. And this often overlooked surgeon from Britain in the 1800s, Campbell de Morgan, you may recognize his name because there are these benign red spots on people's skin that are called Campbell de Morgan spots, and if you're the right generation, apparently Beyonce has quite a few of them. But Campbell de Morgan, amongst other things, was actually a very astute clinician, and he noticed that quite a few of the cancers that he saw spontaneously regressed. And one of the things that he noted was that the spontaneously regression was often associated with another disease that was very prevalent at the time and was killing a lot of people, and that was tuberculosis. And that's an important thing to remember, that tuberculosis, if you had it, can lead to a regression of the cancer. And he also noted, in probably one of the first epidemiological type descriptions, that family members in which there was tuberculosis were less likely to be afflicted with cancer. So, really, what's the inspiration for the immunotherapy that we've come to recognise today? Well, the inspiration is actually this story about this young girl named Bessie Dashiell. So Bessie was 17 years old. She got a hand caught in the seat in a railway car, and she went to see a young surgeon named William Coley. And she unfortunately had a cancer in her bone, similar to that fossil that I showed you, an osteosarcoma. And William Coley was a young surgeon at the time, and he tried to treat her, and she had an amputation below her elbow. The cancer came back, and over a period of a year, she had lots of setbacks, and then she eventually died. But William Coley was so struck and inspired by her story that he went on to search for a possible treatment and cure. So the other important thing about Bessie was, it's in this picture here. So she was very good friends. She was in fact a childhood friend of this young man named John D. Rockefeller Jr. So you may recognize this name because Rockefeller's father was a very wealthy tycoon, and because of the suffering that Bessie went through, John D., as he was known, convinced his father to set up a philanthropic foundation, and from that was born what we now know as Rockefeller University. So what did William Coley do? So one of the things that he did was he tried to look through all the medical records and all the case reports that were described. And he actually went back and realized that there are reports of spontaneous regressions of cancer, and some of those reports were quite often associated with people getting infections. So William Coley, back in the late 1800s, he's now credited to being the father of immunotherapy, made the very bold decision that if infection can be associated with regressions of cancer, then maybe if we gave the patients an infection, that would actually cure their cancer. Now that was very bold, and in fact, so the infection that they used was erysipelas. In fact, that was actually quite a risky thing to do, and many of his patients actually died from the infection themselves. So this was a very controversial treatment. And you can see here from this New York Times article in 1908 that, so you can read the headline, erysipelas germ as a cure for cancer, Dr. Coley's remedy of mixed toxins makes one disease cast out the other. Many cases cured here. So he realized that injecting the live bacteria was actually not a great idea, so he changed his approach and instead used an approach where he injected heat-killed bacteria, mixtures of bacteria, and that came to be known as Coley's toxins, and in fact in Europe it's still available as a treatment. But the problem was that even though he claimed that there were some spectacular results and in fact some cures, there were also some spectacular failures and deaths caused by the treatment itself. And in fact, Ewing, who was his boss at the Memorial Hospital at the time, actually banned it. And the Memorial Hospital is now what we know as the Memorial Sloan Kettering, one of the leading lights in the fight against the cancer these days. So what's interesting is that if you go back to Campbell de Morgan and the association with tuberculosis, we now have a vaccine, not a very good one, but it's still used worldwide to treat tuberculosis, called BCG, which is an attenuated form of tuberculosis. And in fact, BCG is now still used in the treatment of bladder cancer. So why was this approach not pursued further? Was it just because of the terrible complications? Well, I guess it got overrun by other developments at the time that people thought were going to provide much better outcomes. And so here is probably a very ironic image of, this is actually William Coley administering radiotherapy, and one of the proponents of radiotherapy at Memorial Hospital at the time was in fact his boss, Ewing. And so with the discovery of radium and the idea that that X-rays could actually kill cells, there was a tremendous interest in developing radiotherapy. The other development was the development of chemotherapy. And chemotherapy has actually, if you go back to its origins, it actually is quite an eye-opener, because during the First World War there was a chemical weapon used which was mustard gas. And with the outbreak of the Second World War, there was a lot of fear that actually mustard gas would be used again as a chemical weapon. And in fact, there were lots of stockpiles of mustard gas between the Axis and the Allies. So in America, they studied mustard gas. And out of those studies was the discovery that actually if you modified mustard gas and made nitrogen mustard, it could actually kill the white blood cells in your body. And that became one of the first chemotherapy treatments, and it was used to treat leukemias. But as you can imagine, mustard gas is incredibly toxic, and this still is one of the biggest problems with chemotherapy, even in the modern day, and that is that it's a very toxic treatment. It kills a lot of cells, and it kills it indiscriminately. That indiscriminate killing of cells gave rise, I guess, to the idea in the last couple of decades that maybe we should more specifically target the cells. And this idea is rooted in the concept that cancer cells have mutated. They've escaped the normal cellular controls that would normally tell a cell stop dividing and just stay still. Instead, these cells don't obey those cues. They keep dividing rather than staying still. They invade and they spread to other parts of the body. And so these mutations that allow these cells to escape these normal cellular controls normally do it because they change receptors on the surface of the cell or signaling molecules. And because of these mutations, then the idea was that we can specifically target them. So this is one of the first molecularly targeted therapy. It's a molecule called Herceptin, and it targets a molecule on the surface of breast cancer cells, which is mutated in a number of patients with breast cancer, a so-called HER2-positive breast cancer. And this has been quite successfully used in the treatment of breast cancer. But one of the problems with molecular targeted therapies and also with chemotherapies is that, unfortunately, resistance is inevitable, and patients escape control, the patient's cancers escape control, and the disease relapses. So, despite surgery, radiotherapy, chemotherapy, and now molecularly targeted therapies, there have been some improvements in patient survival, but the real problem is that overall there's been no cure. And so you can see here, this is data from Australia, that there's cancer is a problem that's growing, the incidence is rising, but the death rates have only just started to creep down a little bit. And so what we really need is a game changer, something that can actually provide a cure. And that game changer has come in the last few years with the spectacular rise of cancer immunotherapy. So I'm just going to show you some movies that will illustrate the power of the immune system. So here is, this is essentially in a test tube, a movie of a cancer cell, and this is a natural killer cell. So as you can tell by the name, this is a cell of the immune system that's specialized. It's born to actually go out and kill other cells, but normally it's prevented from doing that. But if the other cell is a cancer cell, then it should be able to go ahead and do that. So as the cancer cell dies, as the cancer cell dies, it flashes a red color because that's a dye that's taken up in dying cells, and you can see it breaking up and blebbing. And this can happen in a space of a few minutes. So within our immune system, we have natural killer cells that can very effectively kill cancer cells. So now I'm just going to show you another type of cell that's very good at killing cells called a cytotoxic T cell. So as the name suggests, cytotoxic, it's a cell-killing cell. And so these are movies that were made and published a long time ago now from a group in France where they made the tumour cells glow green when it's red, and the cytotoxic T cell is glow green, yes, and the cytotoxic T cell is red. So there'll be some examples. You can see here's a tumour cell, these are the cytotoxic T cells killing it, and then it will become green. And there'll be a few other examples. So here's the tumour cell that's going to get killed by these cytotoxic T cells there. So this is called the kiss of death, because the way the natural killer cells, the cytotoxic T cells, interact with these cancer cells delivers a death signal to the cancer cell. So our body certainly has the capacity to kill the cancers and cancer cells, so why doesn't it do it? So really, what sets off the immune system? Well, the answer to that came from a really brilliant Australian, Sir Macfarlane Burnet, probably the father of modern immunology, and he won the Nobel Prize in 1960. Amongst his many achievements was this idea of the clonal selection theory. But what this does is it places a context in which we can understand how the immune system works. And the whole idea is that the immune system has evolved to protect us from the outside world. So to do that, it must be able to recognize what is self, our cells, and what is non-self, the outside world. And anything that's non-self is a danger and a threat to us, so the immune system attacks it. And so the clonal selection theory says that there are immune cells would recognize a normal cell as being self and then not attack it. But if you had a cancer cell, because of the mutations within the cancer, then this is a mutated, altered self. It no longer looks like self, and therefore the immune system will think that it's foreign and go ahead and attack, and the consequence is that it would then kill the cell. So how can we use that? How can we wake up the immune system? Well, so you saw how William Coley tried to provoke the immune system deliberately by using bacterial products, his Coley's toxins, and then BCG. Another way to try and provoke the immune system is to actually give it the growth factors that make the immune cells activated and grow and expand. And these are things called cytokines. And there's some examples, interferon and interleukin-2. But remember, this is just non-specifically activating the whole immune system. Then other attempts have been to vaccinate patients against their own cancers using dendritic cells. And then more recently, to specifically target the cancer cell itself using what are called monoclonal antibodies that Stu and Elisa mentioned. So these are very specific proteins made by our immune cells that can recognize very, very specific motifs or signatures on the cancer cells. So this is the first generation of immunotherapy drugs, and I've just sketched together a timeline of what, just to give you an idea of what's been happening in this space. So you can see that Coley's toxins, that was first tried in 1891, and it's not for another nearly 100 years until the second immunotherapy came along, and that was BCG for cancer. In the late 80s and 90s, there was a burst of interest in using cytokines, such as interferon and interleukin-2 or IL-2, and then a bit later, in 1997, the first monoclonal antibody, Mabthera. But the problem with these is this first generation of immunotherapy drugs was that they were very ineffective and they still had lots of problems with toxicity, particularly some of the cytokines because of this non-specific activation of the immune system. So you were getting a cytokine storm that was causing a lot of side effects. And that led to a short hiatus in the interest in immunotherapy. But as you'll see, in the last 3 years, there's been a resurgence in immunotherapy with the second generation. So why is it so hard to kill a cancer cell? Well, this is a really complex area, and one that our lab and thousands of labs around the world are trying to understand. And this cartoon here gives you a sense of how complicated it is. So normally what we'd like to see is that if there's a cancer cell, that it would be eliminated. So your innate and your adaptive immune system would recognize that it's a cancer cell, and these cells, the NK cells, the cytotoxic T cells and other cells would come in and kill the cell. That's what we really want to see happen. But unfortunately, because the cancer cell is a cell that keeps mutating and changing itself, it makes it very hard for the immune system to recognize it. And not only that, in the process of mutating, it acquires strategies with which to evade the immune system. And so you get this sort of equilibrium set up. So what's been described as a Darwinian struggle between a cancer cell and the immune system. So the cancer cell keeps changing, mutating, in the same way that viruses such as influenza to try to escape the immune system. And so eventually the cancer wins and escapes from immune control. So whether it be because now there are suppressive microenvironments so that the immune system doesn't work very well, or whether it's because the immune system can't even recognize the cancer as being foreign anymore because it's developed ways to evade being recognized by the immune system, the cancer cell escapes and grows. So these are the what's happening, we think, in the cancer microenvironment, this struggle between the immune system and the cancer. And so the consequence of all that is that you get suppression of the immune response. And how can we overcome this? Well, this really, everything got turned on its head a couple of years ago with the description of two major immunotherapies that I'll mention, which was recognized, its importance was recognized as being the breakthrough of the year. So cancer immunotherapy was the breakthrough of the year in December of 2013. And so I'm going to tell you a little bit about the second coming of immunotherapy. So I mentioned to you before, a cancer cell should be recognized as foreign and should be destroyed, but it's actually not as simple as that. What we've learned since is that to actually kill the cancer cell, the immune cell needs to be licensed. There's a very good reason for that, because you don't want your immune cells to run around attacking willy-nilly. The consequence of that is autoimmunities, as Elisa mentioned to you before. So these checks and balances are put in place to make sure that the immune cell is appropriately activated and then licensed to then go on and kill the cancer cell. So there's a licensing step, and also there's a checkpoint, if you like, that says, actually, no, don't go and kill the cancer cells. And so the immune cell expresses on its surface these molecules, these co-stimulatory and co-inhibitory molecules that actually can then tell their cell which way to go, whether to kill or not to kill. One of the problems is that the cancer cells have gotten wind of this, and they can actually try to express high levels of these inhibitory molecules, so that when the cancer cell is sitting there and the immune cell comes along, it kind of says, actually, there's nothing to see here, move along. And over a period of time, the immune cell itself can also, because they express high levels of these co-inhibitory molecules, become very sensitive to this sort of trickery that the cancer cells established. So what's turned it on its head in the last few years is essentially really pioneered by this man, James Allison, who's been working for a long time on trying to block these co-inhibitory receptors, what we call checkpoint inhibitors. So what the checkpoint inhibitors do is they come along and they inhibit this interaction so that the immune cell is now able to kill the cancer cell. So this is one of the first checkpoint inhibitors, ipilimumab, and what I'm showing you here is data from Anthony Rivas. So this was so this is America, so it's kind of back to front, not only upside down. So this is April 2011, and this patient received ipilimumab, and you can see that this is a PET scan. All these black spots are metastases from this patient's melanoma. And 4 months later, you can see really dramatic clearing of the melanoma. So this actually points to the power of the immune system. And with the second generation of immunotherapy, the key focus here is to have drugs that actually allow more specific targeting of the cancer cells. Now there's a growing list of cancer cells, not just melanoma, non-small cell lung cancer, head and neck cancers, bladder cancer, stomach cancer. I mean, the list is growing as we speak of drugs that are actually treatable with these checkpoint inhibitors, indicating that these drugs may potentially be a drug that can be widespread use. The other exciting thing is that it's not just CTLA-4 and PD-1. There's now a growing list of checkpoint inhibitors. So this space is growing very fast. It's becoming incredibly complicated. But the key thing here is that it's very exciting because there are more and more drugs that we can actually try in the clinic. So the other key advance is the development of this technology called the Chimeric Antigen Receptor, or abbreviated to CAR T cells. And it's thought to be a way to turbocharge the immune system. And so what this does is it takes the monoclonal antibody that was used previously that could target the cancer cell, target something that expressed very highly by cancer cell, and then they hook it up to the T cell receptors. Remember those cytotoxic T cells that can kill cancer cells. So now you have a cytotoxic T cell that now has this receptor that recognizes the cancer cell. But on top of that, it takes advantage of our discovery that not only do you need to recognize a cell as foreign, you need to have this co-stimulatory molecule. So they've now hooked onto it 1 and now 2 co-stimulatory molecules so that the cytotoxic T cell not only can see the cancer cell, it's getting that green light to go ahead and kill it. And that has been, involves a little bit work. So the patient's own cells are taken from their body and they're engineered to express this very clever chimeric antigen receptor and then infused back into the patient. And in this example, the chimeric antigen receptor is recognizing a molecule CD19, which is expressed at high levels on lymphomas. And so then this will be used to treat a patient with lymphoma. And as you can see in this example here, here's another PET scan, and here's a large mass in the kidney of a patient with lymphoma before and then 2 months after treatment with the CAR T cell. So people are now seeing very spectacular results to the point where now my oncology colleagues are saying they're even comfortable enough to use the word cure for the first time, that they're telling their patients they're actually being cured. So this is a really exciting and a great time to be studying cancer and the immune system. But this slide is really important because it really not only tells you the impact of immunotherapy, but the challenges ahead for us. And so here plotted on this is a sort of like a schematic is patient survival with 100% up here and 0 down here, and months since diagnosis. And you can see that with chemotherapy, you can prolong patient survival. With molecularly targeted therapy, there's been some improvements in survival. But the key point here is that patients still die. And that's the point that I want you to remember, that immunotherapy for the first time is curing patients. So that now, rather than this survival curve going down to here, it's flattening out, it's plateauing. The other key point, though, is that even though immunotherapy is curing patients, it's not curing every patient. So at the moment, immunotherapy is only used in a small number of cancers, and in the cancers in which it's used, not everybody responds. And that's really one of the great challenges ahead for us, and that is to work out how we can improve the responses in immunogenic cancers, and then take other cancers where they're not so immunogenic, where there aren't so many mutations for the immune system to target, and convert them into an immunogenic cancer so that we can universally treat all patients with immunotherapy. And then I guess my last and probably most important slide is actually just to thank you for your attention. I'd like you to remember Bessie Dashiell and the impact that she's had in getting us to where we are today, and then just to remember that your support makes a huge difference to what we do here at the Garvan. Thank you very much.